Davide Sassera from the University of Pavia and the University Hospital of Pavia. Today I will tell you about work that we're currently performing and our preliminary results on sweep-metagenomics to investigate the variability of Candida within our hospital. Why Candida? Candida is an important fungal pathogen. Probably you know about Candida auris, it's the most famous one. Luckily, we don't have Candida auris in our hospital, but we do have some important species that can cause multiple kinds of diseases, including the most important one, candidemia. And this is also important because of the rise of isolates that are resistant to antimycotics. So we know a lot of bacteria can be resistant to the drugs that we use against them, but this is also true for fungi more and more so. And the way we try to investigate the diversity of Candida in our hospital is by using sweep-metagenomics, which is a novel method that we developed thanks to the main effort by the UK Corander group in Oslo. And the idea is to have a method that's more powerful than single colony genomes because it can see the diversity that's present in a clinical sample, but it doesn't have all the issues that can be present in shotgun metagenomics, just to put it briefly. So what did we do? So we select a cohort of over 200 patients and we subjected them to the sweep-metagenomics approach. And we were able to obtain samples from these patients in a longitudinal scale, let's say, so that we were able to follow them for days. And we collected rectal swabs, we collected nasal samples and respiratory samples. Then we subjected them to sweep-metagenomics and we obtained over 600 positive sweeps. With the news bioinformatic pipeline, there was a ad hoc design for this kind of isolates, and we were able to investigate the diversity that was present in our hospital at the ward level, but also within at the patient level, because we were following them. So what did we see? We found that a couple of species were the most common one, albicans and parapsilosis mostly, and we were really interested in parapsilosis because we already knew there was a fluconazole resistant isolate that was present. And currently we want to investigate this diversity below the species level because the method is very sensitive and will allow us to, let's say, divide these species into what's the equivalent of sequence types for bacteria. And in parallel, again, I was saying this is very preliminary results. What we can see is we can follow patients. And you can see here in this patient that we have multiple types of samples. And I will focus on the triangles, which are the rectal samples. And you can see that at the beginning, this patient was colonized by parapsilosis mostly, with a slight bit of Candida albicans. But towards the end of the study period, Candida glabrata appeared, and then it became prevalent at the end. So the fact that, of course, we have whole genomes for all these bugs will allow us, when we finish this work, to try to understand why one strain from one species can displace another one, and hopefully we'll better understand things like what are the mechanisms of resistance, because this is really complex in Candida. There's like SNP variations, copy number variations, aneuploidy. And so it's an exciting data set that we're currently analyzing. And lastly, I would like to thank the people involved, especially Michela Vumbaka. She is our PhD student that's mainly working on this, but also all the people at the university and the hospital who helped with this. Thanks.